Welcome to our Turing-Roche Knowledge Share series. The aim of these monthly events are to bring together members of the Turing and Roche networks and the wider scientific community to hear partnership updates, different perspectives on topics in data science, and also build new connections. So just to get us started, I'm just going to give a quick overview of the partnership. Hopefully you can all see those slides. So the clue is in the name with the Turing-Roche Partnership. It's a partnership between the Alan Turing Institute and Roche. I'm sure most of you will be familiar with at least one of these organizations, but just some background information if not. So the Turing Institute is the UK's National Institute for Data Science and AI, established in 2015. We're a convening body of university, industry, charity, and government partners. And the Turing-Roche Partnership sits under our Health and Medical Sciences program, and Roche is Turing's first pharmaceutical partner. Roche is a global healthcare company with a long-term interest in personalized healthcare, and they also have a growing number of academic partnerships. And together, we launched our strategic 5-year partnership in June 2021. And this is our kind of partnership North Star, so our guiding aim for the 5 years. So ultimately, we're collaborating to explore this term, patient and disease heterogeneity, for advanced analytics. So why patients respond differently to treatment and why diseases manifest in different ways in different people. Obviously, this is quite a broad aim, so we're tackling this through a stepwise iterative manner, which you can see illustrated on the right, with a main focus on developing new methodology, and focusing on different research themes such as missing data, predictive modeling, and multimodal data integration. And you can actually find out more about this on our Turing webpage, which is linked to there. Our core partnership team is pictured here with representatives from Turing and Roche. And then this is also expanded out with researchers we have funded, Turing enrichment students we've taken on, and our community scholars cohort, which I previously mentioned. Again, you can find out more information about this on our webpage. And also a key part of the partnership is wider connections and collaborations we want to build. So it's really critical for us to be running events like these to get you all involved. So our theme today is digital twins in health. The concept of digital twins has been around for a while, particularly in engineering, but there has been recent interest in how these can be deployed in the health space. So I'm really excited to hear about that today. So I'm delighted to welcome our speakers, Steve Niederer, who is Chair in Biomedical Engineering at Imperial College London and co-director of the TRIC-DT, which is the Turing Institute's Research and Innovation Cluster focusing on digital twins, and also Jon Walsh, who is the co-founder and head of modeling at Unlearn.AI, who is working on a project that he's going to tell us about with Roche colleagues. As mentioned, we'll be hearing from both our speakers, and then we should have plenty of time for questions at the end. Feel free to ask specific questions to an individual speaker, or you can ask a broader question around digital twins for both of them to answer. And with that, I will hand over to you, Steve.
Excellent. Thank you, Vicky. Thank you for putting this together. And there we go. So the presentation I'm going to give today is kind of talk about where we are with making digital twins, and specifically in my group, we work on making cardiac digital twins or digital twins of patients' hearts. And thinking about this kind of Roche Turing initiative, it's looking at heterogeneity, and we're particularly interested in why patients who receive the same guideline-directed therapies have such different results, and how can we use digital twins of individual patients to tailor results and to make sure that we're getting the right diagnosis for those individuals, getting the right therapy, and then when we're giving that therapy, making sure that's delivered in an optimal way for that individual, so that we try and get the best result for each of the patients. So I would say that with the idea of making digital twins, when we think about engineering, where you might be looking at making digital twins of an aeroplane or a refinery or a large static resource, is kind of contrasted with healthcare, where we're having to make digital twins of large numbers of peoples and large numbers of entities. And so this really poses some unique challenges. So we start off with kind of what is the vision. And so with our digital twin, we would start with our patient who has their heart here. And if we think of conventional healthcare, they might come into the hospital, have a diagnosis, and have clinical decisions. And as we kind of grow the field of resources around Internet of Things and wearables, there might be continual monitoring and then some type of feedback from your Apple Watch or whatever other wearable you might have. I think if we start thinking about how are these connected, so sometimes the wearable information is available to healthcare, but often that's still an area of development. And then if we think about where a digital twin comes in, we start to see having a digital twin where we can start integrating this information from both the hospital and wearable devices, combine that with population priors as well as physiological priors and physical constraints, start making a mathematical representation of that individual patient, in this case of their heart. So we can better look at how we can better monitor those individuals, better forecasts, run what-if testing, so if we give this therapy or that therapy, how do we think they'll respond, and provide greater insight. So this is where we focus on the personalized care. And I think the other area in health that's going to be interesting is the idea of digital networks of digital twins. Where instead of just one twin with an observed patient trajectory and patient forecast, we have large numbers of these twins. And how do we start linking these together and sharing information between the digital twins across this network to kind of make more resilient, scalable, and accurate patient digital twins? So this is the kind of roadmap that we're working towards. However, this is very much a roadmap that we're at the beginning of. So what do a mathematical model of a heart look like? And so this is a multi-scale and a multi-physics problem. So we start off with, in our case, making models of electrical activation. So every heartbeat, the heart has an electrical signal that propagates over the top two atrial chambers before activating the ventricles, the bottom two chambers of the heart. This electrical signal is represented at the cellular scale by ion channels, which are representing the kind of individual proteins, which are carrying ions in and out of the cell, regulating that electrical signal at the cellular scale. This electrical signal at the cellular scale gets converted to a chemical signal in the form of calcium that's released from inside the cell. Calcium binds to collections of proteins called the sarcomere, which generate tension inside the cell. This tension is developed inside a tissue matrix, which has a preferred orientation and stiffness. We can combine that with measurements of patient anatomy to then start to simulate whole heart mechanics, and combine this with models of computational fluid dynamics to simulate the blood flow in and out of the heart. So this provides us with a comprehensive system which goes from the whole heart electrical activation, which is something we can measure with clinical diagnostic things such as the ECG, through to the mechanics and blood flow that we can measure with echocardiograms or MRIs or CT imaging devices. But it does this by linking it through all of these different molecular scale and cellular scale properties, which is where we're seeing diseases develop, genetic or inherited cardiomyopathies be represented, as well as whereas a large number of drugs are going to be operating. So it really provides a system for linking this molecular scale through to the whole heart. And so we can use these models to make simulations of large numbers of outputs that we might see, such as the pressure transient, how much volume is being generated, pressure volume loops, which provide a representation of work in the different chambers of the heart, and representations of whole heart electrical activation. And one of the challenges we have then is to link these cellular properties that we might have to these whole organ simulations. And just as some calibration, this whole heart simulations are taking about 7 to 8 hours on a 500 to 700 core machine. So each sample is exceptionally expensive. And so we might want to look at how the atrial myocyte affects the whole heart function, the ventricle myocyte, the cardiovascular system, which provides the blood flowing in and out and regulates the blood flowing out of the heart, the tissue properties in both the chambers, the differences that we might see, heterogeneities we might see in how boundary conditions which constrain the heart might affect it. So this is where we're starting to look at using large multi-scale global sensitivity analysis, where we can do large numbers of simulations, train emulators or machine learning, low-cost representations of our model to make a map from the input parameters, which we have along the bottom, which are all 42 different parameters for this model, and all of the different clinical indices of mechanical function that we might be able to measure from MRIs or CTs or pressure catheters. What this allows us to do is effectively have a sensitivity map where we can say which of the parameters on our x-axis have the greatest influence on the output parameters on our y-axis. And we can see the circulation has a big impact, but we can also map these model parameters down to genomics and particular genes. So this provides us with a map of saying how can we look at how particular proteins might influence whole organ function that we can do with these multi-scale models. Another area of interest for us is, and again, thinking about this Roche heterogeneity problem, is where we have multiple therapy options and how do you choose the right therapy for a patient. So if we think of the healthy heart, this has a rapid activation in both the left and the right ventricles. One of the disease that exists is left bundle branch block, where the rapid activation on the left side of the heart fails. And so we have a slower activation coming from the right to the left side, and this leads to a desynchronous, inefficient contraction. And what we can do is we can pace the heart with wires and devices which are implanted in the heart to return the heart to synchronous contraction. So there's conventional, what we call cardiac resynchronization therapy, where wires are placed in the left and right side of the heart. There is endocardial CRT. There is options where we can place the conductance system itself at different locations, so left bundle placing or left bundle placing. And the message here is largely that while we have one disease, we have multiple different ways of delivering therapy to these patients. So how can we identify what are the best therapies in each individual? So this is where we're starting to look at making computational frameworks to start to explore how variability in patient anatomy and function gives rise to variation in response. So we can make a cohort of patient anatomies from heart failure patients at St. Thomas's Hospital. We can generate a model of the rapid electrical activation system, which is going to both fail and potentially be paced by these devices. We can then do simulations, and we can measure multiple different values that people have reported for response. So every clinical paper, different groups will report different indices, and sometimes it can be very hard to compare results between different groups. And so using a simulation, we can report all of the different indices that have been reported by all the different groups. And again, it's a way of addressing some of that heterogeneity we might see in the data. And this allows us to do what we call an in-silico simulated clinical trial, where we can start to simulate how multiple different devices delivered in multiple different ways to different patients might lead to different outcomes. And here down the bottom, we can look at the activation maps, where we'd like these to all be as yellow or red as possible, as synchronous as possible. And we can see that for these particular groups, that the left bundle branch pacing has a rapid activation on the right side and a slow activation on the left, and that this gets recovered the best with his bundle pacing, which will be kind of the best therapy that we can see for this patient group. We can also think about how we might need to deliver therapy optimally in each individual patient. So this is a different form of this resynchronization therapy, where an electrode is placed on the endocardium of the patient's heart, and it's powered by an ultrasound transducer, which is placed in the intercostal spaces between the ribs. So for each heartbeat, an ultrasound pulse is sent by the power transducer, and that goes in to power the electrode. And what we'd like to do is work out where best to place the electrode and the transducer in each individual patient. And can we use modeling and simulation to do this to predict where best to deliver therapy? And so this again requires building these kind of systems for trying to identifying where best to pace or where to deliver optimal therapy. So we have motion models for identifying where the last contracting region is, to see if we can identify the late contracting regions, which are pacing targets. We can look at perfusion measurements from interpreting cardiac CT images. We can look at wall thickness measurements to make sure that when we put the electrode in, we don't puncture the heart. And we can look at doing simulations to identify where the best place is to put the electrode to deliver a synchronous activation pattern. And so we can make these models for each individual, and we can do this within the clinical workflow. So currently patients will come in, they'll have a CT, we'll do our analysis. Then during the procedure, we will register the results of that model with the imaging modality that they're using during the procedure, so we can put the results of our model in front of the cardiologist to guide them in real time. And then we can provide them with these target regions of where they want to put the electrode, and they can then move their catheter to put the electrode on this location. And this allows them to identify the optimal place to put the electrode to get a best acute hemodynamic response in these patients with this device therapy. Then the other area we've been looking at has been how we start scaling this up. So these have been ideas about making models of small numbers of hearts, but can we apply these to large cohorts? So UK Biobank being a fantastic resource, which has a large number of heart MRIs, about 56,000, and a corresponding large numbers of ECGs. What we wanted to do is to start to test, can we take these approaches that we've been applying to patients where we have very rich data sets, where we've been able to control the data collection, control the analysis workflows, but apply these to large population cohorts. And so What we did was we looked at the MRIs and how we could automatically segment those MRIs to build anatomical meshes representing each patient's heart or each participant's heart. We then wanted to see if we could then simulate the electrical activation pattern in these patients' hearts, and then use that electrical activation pattern to then simulate the ECG recording that would come from that patient's torso. And then we can then get an estimation of the cure restoration from the simulation. And what this does is this provides a framework where we can go from the MRI, which is measured, all the way through to predicting the ECG from the MRI using a physics-constrained mathematical model of the patient's heart. And in doing this, this allows us to tune or be able to tune the material properties of this patient's heart. So we can take a simulation, run all the way through, estimate what the cure restoration is, which is a measure of how fast the heart activates. We can then compare that with what was measured clinically. And depending on how close we are, we can then adjust the material properties of our model to get us a closer estimate of what that cure restoration is, allowing us to combine the MRI and the cure restoration, which are two organ scale observations or clinical observations, to infer a tissue level property, which is going to be regulated by particular proteins such as gap junctions in the sodium channel. So then we want to see, how do we then scale this? And so we can now take our UK Biobank MRI and ECG. We can then calibrate and estimate our conduction velocity from our simulations to estimate the cure restoration or match the cure restoration in each patient. So we now have an estimation of conduction velocity. And so we can now do this in about 8 minutes per case to get the anatomy. We can do this for less than 15 minutes per case to get the conduction velocity, and allows us to build about 100 cases per day. And while 100 cases per day is a very large number, it still means that for 50,000 cases, we're looking at over a year of time on one machine. Luckily, with more money, you can buy more machines. This is kind of representing the scale of the problem for applying this to a large number of patients. And what we found interesting was that when we started to look at this approach and moving from making computational models of one patient to making computational models of thousands of patients, we can start to ask different questions. And so this is obviously a busy slide, but we can look at the red, which is the cure restoration, the conduction velocity, which has been estimated, which is yellow, and the myocardial volume, which is purple. And we can start to look at how changes in potentially the cure restoration are represented in male and female hearts, for example. So we know that we see here there's a significant difference in the cure restoration between male and female hearts. Now, is that because we have a big heart, which takes longer to activate in men? Or is it because the material properties of female hearts and male hearts are intrinsically different? So from our analysis, we found no difference in the material properties between the male and female hearts, but we did find that the entire difference in the ECG could be explained by differences in the myocardial volume. So it provides a way of us starting to explore and start to unpick some of these differences that we might see between different groups, such as male versus female, and start to say, is this because of anatomy or is this because of intrinsic material properties? And we can use similar approaches to explore how myocardial properties might change with BMI, as well as how it might change with age. So for us, this is an interesting way to start to explore how, if we can use models to identify these differences between different patient groups, and also how this might then lead to differences in the response to different therapies or different drugs. So what we're really trying to do is looking at how we can start to make a computational model of an individual patient's heart, and how we can then make this model available to our clinician here, how they can play this heart and look at it, how they can adjust it to return their sick heart to a healthy heart. And this might be a doctor and also might be a patient looking to explore how they can improve their cardiovascular function. But it's about making these tools available. And this was just to recognize different people who have contributed to this work, including my current and past postdocs who've gone on to different roles. Thank you.
Thanks so much, Steve. That was great. Any questions for Steve can go in the Q&A box in the bottom bar, and we can get to those after we've heard from Jon. So Jon, take it away.
All right, thank you. So I'm excited to be able to tell you about a different application of digital twins, one that's more targeted towards clinical trials. So this is collaboration work with Paul Delmore and Spiros Rupinis from Roche. And in this case, we're really approaching digital twins for human health at the level that you measure data from humans in clinical trials. So on the right, you see an example of patient record you might think of recording during a clinical trial. You have some measurements at baseline, the data you take at randomization for the trial, and you ultimately want to know, and you're going to measure the outcomes of that patient during the trial along a number of different directions. So these could be, for Alzheimer's disease, for example, cognitive, functional, quality of life assessments, neuropsychiatric assessments, labs, vitals, other safety information. And we think of a digital twin in this case as a comprehensive, computationally generated probabilistic prediction of a patient's outcomes. And the patient outcomes that are being predicted are outcomes on a standard of care. So in a trial, we're testing a novel therapy against some kind of standard of care, and we want to understand, is that therapy more effective? So the digital twins are a tool to build expected outcomes of patients under that standard of care and improve the statistical properties of the trial to make it more efficient, more powerful. So I'll talk today about how we build digital twins and then a particular application in a case study in a clinical trial. So I really want to think about, for each different outcome I might measure, as the digital twin is representing some kind of potential outcome of that patient under different scenarios that I might realize. So there's a lot of environmental noise in human health. And so when I measure an outcome in the future, it's not just one value, but a distribution. And the digital twin model is going to reflect that. So I can think about at any time the patient's potential outcomes as a distribution. And the goal is to learn from data from past clinical trials and other historical data, the distribution, the multivariate distribution of that data that I'm going to measure. So I can think about, in practice, what's going to happen is I take data during a clinical trial. At randomization, I don't know which arm the patient's going to be in. I don't know any follow-up data that I'm going to use to analyze treatment effects. I'm going to put that data through some kind of machine learning model that has learned the representation of data over time. And that's going to predict the outcome of the patient under standard of care. And that's their digital twin. So it's some prediction over many variables of the likely outcomes of that patient. And these are, they have an associated mean prediction and a confidence or, an underlying distribution associated with that patient. So I want to take just a moment to talk about, what kinds of models we use to do this. They're different, they're physics-informed models in the sense that they're using machine learning models based a lot from things that have been developed in physics, but they're not at the same level of detail that Steve developed because they're talking about the whole level of human health. And they're really targeted at what properties of clinical data you often encounter in the wild. So clinical data is longitudinal. It's multimodal, meaning there are different data types that are represented. This can include, features from images or other potentially novel therapeutic mechanisms, continuous, binary, ordinal data. Missingness is extremely common. So I have lots of cases where I don't know the data that was measured because it wasn't taken for various reasons. And I don't have lots of data. So I may have thousands of records of patients in past trials, but not millions like you have in electronic health records or other applications. And then the conditions in which I take data, the actual studies in which data is taken tend to vary. So all of these kind of add up to a very different data landscape and machine learning problem than you typically encounter in other, industrial AI applications like images or text or things like that. So to handle this, we've built a class of machine learning algorithms tuned for clinical data. So the general area is probabilistic neural networks. One particular architecture we've used a lot is restricted Boltzmann machines. And they're a latent dynamical model where you have a set of visible variables that I measure, the variables I'm actually interested in modeling. And I have a set of latent variables that are modeling the dynamics of the relationship between those variables. And I can train these models and they really handle a lot of these challenges well. They're not commonly used in a lot of other AI applications, but they're really well suited to a lot of the properties of clinical data. Okay. So when you think about using digital twins in trials, you might be really concerned that the decision about whether or not a treatment is effective is really subject to potential bias. And what I don't want to do is bias the decision about is this drug effective or not, because with models that haven't been rigorously tested in the field and in trials currently. So the regulators, FDA, EMA, think of this as a context of use. How you use digital twins, how you use machine learning models in trials is really important to think about. And we spend a lot of time thinking about the applications of digital twins to make treatment effectiveness decisions more efficient. So ICH, one of the governing bodies for the rules in which clinical trials are conducted, that issues guidance, has this one guidance on how you analyze clinical trials. And one of the important features of clinical trials is you can adjust for covariates because you have randomization. So randomization, when patients go to both the treatment and control arm randomly in a blinded way, provides a statistical cover for being able to account for covariates and things that can affect that outcome in ways that you can robustly account for in the analysis. So, ICH and FDA recommend that you should identify covariates that are expected to have an impact on the primary variables you're interested in and adjust for them in the analysis. So under the ANCOVA model, you can adjust for prognostic scores and other information you believe is going to be able to associate with the outcome, because doing so will lead to a smaller uncertainty in your treatment effect and more precise trial, more powerful trial. So this is the framework that we use digital twins in for phase 2 and phase 3 trials. We've published on this in the past, and this is a method called PROCOVA, prognostic covariate adjustment, where you use a complex model applied to pre-randomization data to produce the optimal covariate for adjustment. And you can prove it's mathematically optimal under certain basic assumptions. And we've gone through interactions with FDA and EMA. EMA has a European Medicine Agency has a qualification program where they've qualified PROCOVA for use in phase 2 and 3 trials to add power or to make sample size reduction. So this is a framework in which it's been well tested, reviewed by regulators to meaningfully apply AI models and twins in clinical trials. And ultimately, the goal is to improve randomized control trials and make them better in various ways. So I think about my basic representation of a trial as having a treatment arm and a control arm. Oftentimes the randomization is 1-to-1, and I have different designs in which I might want to add more power. I might want to, so the digital twins being added to the trial, to the analysis of the trial, adds more power to that study. I can use that power to enroll fewer patients, so I can recover some design power and get a smaller total enrollment. I can instead spend that power to have a smaller control group, which I require fewer patients to be taking standard of care. And I can even try to optimize the randomization ratio so that more patients, the maximum number in my trial, have a chance to get the treatment. And so you can use digital twins with these statistical analysis methods to optimize the design of trials, and that's really the goal. So the collaboration with Roche was oriented around evaluating the quality of digital twins using a previously completed clinical trial. So Roche conducted the ABBY study in Alzheimer's disease. It's a phase 2 trial from 2011 to 2014. There were 18 months of follow-up and a total of 431 participants. There are 2 endpoints to this trial. So there's a cognitive assessment that's given to patients that is called ADAS-Cog12, and also a functional assessment called CDR Sum of Boxes, CDR-SB. And those were the primary endpoints of the study. So we use data from the placebo arm to create digital twins and compare them to observed outcomes. So the basic scheme is to take the placebo data from at baseline, to feed it through the model that we've built to create digital twins, and then compare that to the placebo outcomes and do a statistical analysis on the result. And so for ProCova, for this approach to use the digital twins as a covariate, the thing that you really care about is the correlation between the predicted outcome from the digital twin and the observed outcome. The baseline score is often used as a standard covariate in analyses, so it's the value of the particular endpoint that you're looking at the time of randomization. And the endpoint is the change of that score over time. And so for the baseline scores, the correlations are small, and it's typically fairly true. For single variables, you often get very small correlations. But when you build this digital twin and combine as much data as possible, the correlations you find are much larger, and this allows you to have significant impact then on the design of future studies. So if you translate these performance numbers to improvements in these different designs, you can take the power up significantly from 80% to 85 to 88%. You can reduce the sample size, reduce the size of the control arm, and also have significantly better randomization ratios that allow more patients to be randomized to the treatment. So this kind of approach is the kind of approach that you use to validate. You can use to validate these methods and determine how you want to design future studies using past data from previously completed trials. Thanks.
Thank you so much, Jon. Very on-time talks, so we have plenty of time for questions. I can see a couple have come into the Q&A chat already. You're also very welcome to raise your virtual Zoom hand via the reactions button if you'd like to ask your question live. If not, I will get started in the Q&A box. So the first question for you, Steve, is when modeling the forward problem of cardiac electrophysiology, so from cardiac source to ECG simulation, not only an anatomical model of a heart is needed, but also a model of a patient's torso and the orientation and location of the heart in the torso. How would you approximate this from clinically available data, as torso MRIs are not generally available?
No, so this is a very valid question and a challenge. So what we did for UK Biobank, where we didn't have the torso, we obviously had a representative torso, and we could then do a sensitive analysis to look at which were the features, what were the parameters which had the biggest effect on the ECG. Was that the location of the ECG electrodes? Was it the location of the heart? Was it the orientation of the heart? Or was it the material properties? So that was how we were able to kind of narrow it down to have some confidence that the material properties we were predicting were the most important, even though there was a reasonable amount of uncertainty that was introduced by the lack of knowledge of where exactly the heart was. Certainly in the hospital, where we have more control over that data, we can then get the navigational images from MRI, as well as some wider images from CT for constraining those hearts. And I think if we were doing this kind of moving forward, there is obviously the ability to start to infer from the limited amount of information you have what those locations of torso might be from imaging completion approaches. So that would also be something to look at, obviously with uncertainty.
Great, thank you. Next one for Jon. If we're interested in generating novel evidence for a clinical trial, identifying novel treatment effects, where do you get training data for the machine learning model that generates digital twin in a given trial?
Yeah, so this is fundamentally a similar question. How do you get data to build these models? So we've used data from past clinical trials, observational studies, and other sources. The challenge for building any of these models is you need past data to understand the disease progression of a similar patient population. The goal is to model progression on standard of care, not to model the actual novel therapy being tested. And so for many disease areas, there's an ample amount of data that's been collected in the past. And that's the kind of data we collect and curate, and that's a lot of the work actually in building the models.
Great, thank you. Next one for Steve. What would you say is the next possible necessary step in the process of upscaling cardiac digital twins for routine clinical use?
So I think it's identifying strong use cases where there's basically clinical need, where there's such as a problem that the clinicians are struggling with, and then there is a pull from the clinicians to adopt it. I think if we go in there and from a top-down approach and try and provide it as a service initially, that will struggle. But there's certainly a number of small exemplar cases. We've been looking at cardiac synchronization therapy where devices are implanted. There are patients with pulmonary arterial hypertension where smaller patient groups who have a strong need for better care. And these are some of the groups that would certainly be targeting. So I think identifying those groups and then developing the models and having the clinical buy-in to demonstrate those in practice is really the next step for us.
Great. And I think actually I'll go to the next question, I'll come back, but I think this is kind of a broader question around, do you think kind of digital twins can be the simulator for AI machine learning models before the application on the real humans? So I guess that's kind of what you were saying, Steve. I don't know if there's anything else you want to add.
Yeah, I certainly think that the either training, the simple ways where the digital twins can add additional information, which might be used for training or making predictions for individuals, but also for providing kind of augmenting data sets to provide more large numbers of training data sets, particularly for rare events that you might have a very limited number of clinical cases for. You can use simulations to make much more robust AI algorithms. Yeah,
great. And I guess, Jon, that kind of feeds into the same for you, that these are clinical trials on humans. So this is aiming to kind of, have an additionality that helps.
Yes, yeah. So I think, like Steve says, you can use this as a mechanism to try to develop AI tools that can better capture the kind of health outcomes you want to model.
Perfect. And someone's asked for you, Jon, what the results from that clinical trial were. So I guess that was the Alzheimer's one you showed.
Right. So Roche has published, they conducted the study long before we collaborated, and they've published the results. It is in a paper. If you Google the AbbVie clinical trial, the compound, unfortunately, wasn't shown to be effective in the trial, but Alzheimer's is a very challenging therapeutic area.
For sure. Ashish, I can see you put your hand up. You've also asked one in the chat. I don't know if you just want to ask you a question live.
Yeah, thanks, Vicky. Great talks. So my question was to Jon. So is it modeling just the effect of the control arm? And if that is the case, how do you really measure the effectiveness of a novel therapeutic treatment, for example? Because let's say you have two clinical trials taking two very different therapeutic approaches. If the digital twin model is just modeling the control arm, it would do the same thing in both the trials.
Right. So you are trying to model the outcomes of the patients in the standard of care, the control arm. The intent is you can use those kinds of models in many different ways, and that's why the context of use is important. If, for example, you had a very accurate model that could predict the patient's outcome on standard of care, you wouldn't even need a control arm. You could just run patients on the treated arm and compute the difference between their outcomes that they observe in real life and the predicted outcomes from the placebo arm. However, that is subject to a lot of bias. If the model is at all wrong, you have no way to tell that the model's wrong in that trial. And the FDA and the EMA have kind of issued guidance on this. And so how you incorporate the twins becomes really important if you want to run trials that you can then submit to regulators as evidence that the drug works. And that's why modeling the placebo outcomes and then using them in a covariate adjustment analysis is is the approach that we've developed. The goal really there is to provide additional information about every subject. So every single subject in the trial gets a digital twin, and that additional information from the model's predictions is additional data for the statistical analysis to use to get a better estimate of the treatment effect. But it doesn't rely on you having to know anything about the treatment itself. You can apply this then to any new clinical trial without specializing to the actual compound being tested.
Just a quick rejoinder, like often with new drugs, it's hard to model efficacy or safety, for example, right? Like it could produce the desired therapeutic benefit, but it could also have adverse effects, right? So till you actually do it, like if you were to do it without any control arm, then how do you really measure, whether you wouldn't have seen those adverse effects in a control arm, for example?
Right. So it's one of the challenges you need if you were to do a single arm trial, which in certain disease areas where it's unethical to give patients a placebo a trial, that happens. You just need a model that can cover the safety outcomes that you're interested in. There are other approaches to this. There's historical data matching, so using synthetic controls and a propensity score matching to try to do this. But in any case, you need a reference for how much you expect adverse events to occur, how much safety impact there is, things like that.
Yep.
Thanks so much, Jon. Also, next one in the Q&A box for you. So could you please share how was the model performance comparing the virtual data and the real data in terms of time series features?
Yeah, so we're really focused in this work on the correlation between them because that's what's relevant for the analysis. So that one of the virtues of the covariate adjustment analysis is the model can be biased on individual predictions, so you can be wrong, but that bias does not impact the bias of the treatment effect. It's the magic of ANCOVA and randomization. We have not published on the results of the study yet. We talked about this at conferences, but I can't release the information. The data is available within Roche if you talk to Paul and Spiros.
Perfect. I think they're both in the Turing-Roche Slack as well, if you want to ping them. Great. So I think this is probably kind of a question for both of you, but how do we kind of utilize the digital twin model here to help doctors and patients with a kind of decision-making process? Jon, I guess in your case, kind of the decision-making process is around clinical trials, and kind of what levels of thresholds do we need, and any kind of examples around that? Steve, you can kick us off.
Yeah, I mean, I guess it's the same as any decision support tool. And I guess there have been a lot of risk scores that have been available, and they can be very simple, they can be more complicated. And those are evaluated in randomized controlled trials. And if the outcome is better than the other, and you've obviously met all of your kind of quality control for your software, then you would be in a place to do a regulatory submission of that. So I think that while it is very sophisticated and is very different, it's also, there's kind of very complicated risk scores already available that have gone through, and that randomized control trials will be the invariable or the most likely way in which these will be evaluated. So that's how I see them kind of moving into kind of mainstream clinic. At the same time, I see there being a softer entrance to impacting patients' lives through areas in the regulatory space, which has a little bit more population average, so you don't need to be 100% right. Not to belittle the effort, but that there's a little bit more wiggle room with a population rather than an individual. And so I think for kind of helping people do better design of medical devices, doing drug toxicity, drug efficacy tests, bringing some of that information early in the drug development pipeline will hopefully make some of those kind of technologies more efficient, kind of bring many of the modeling and simulation tools that we've seen in aerospace and civil engineering and automotive engineering into medical device design and making more use of that.
Right, yeah. Jon, I feel like you already touched upon this, but was there anything you wanted to add?
No, I think what Steve said is absolutely right. That we started clinical trials are a population level measure. We started with that because it's a place you can apply these types of technologies safely and not bias patient decisions. And as you think about moving out into clinical decision support, where you'll be making decisions about the care of individual patients or small groups of patients, you really need to be mindful of the risks to those patients. I mean, there's a responsibility, right, to not adversely impact patient care, and to also work with clinicians to integrate this into decision making in ways that they can understand and leverage.
Great, thank you. And also, again, I think kind of a question for both and kind of in a similar vein. So kind of what barriers have you identified in terms of adoption of digital twin technology in actual clinical settings? And if so, how are you kind of addressing these?
Yes, I think that we've found that for the current generation of models that we're making, or digital twins that we're making, that we need a large amount of data from each individual, and that the cost of acquiring that data, both financially to the healthcare system and to the patient, is high, and the computational costs are non-negligible either. And so there is very much a scaling question about how do we make models with less data, how do we make models that can be updated with less computation, how can we ideally leverage cohorts of models so that the marginal cost of each additional patient who we want to include in this process is cheaper to do. So I think those are some of the challenges that we've identified and are trying to address.
Great, and then Jon.
Yeah, I think the clinical trial space, people have designed trials for. It's been a fairly consistent set of designs for a long time, and the way that people think about taking data in trials is for the reasons why data is taken is to estimate efficacy in the future, to measure efficacy in the future. Data taken in clinical trials has a long way to improve to try to adapt to technologies like this and more innovative designs. So I think there is this long-term, multi-year, multi-decade kind of revolution that needs to happen of why do we take data in clinical trials, and what is the purpose, and trying to take data that's going to help optimize the ability to make good treatment effect decisions. The types of technologies we've talked about today are very novel. They're coming to spaces that have long established practices, and so reckoning with that takes time, and it's going to take time for approaches that are going to help patients to kind of diffuse out into standard practice. Some of this just takes patience, I think.
Definitely. Steve, another question for you. Do you believe that the use of AI models, so deep neural networks, as surrogates for physics-based models can be valuable in upscaling cardiac digital twins?
So in short, I would say beyond valuable, I think it's going to be essential. That many of the models we solve are subtle variations, or simulations that we perform are variations on simulations we performed previously. And when we have a new patient, we perform a whole bunch of simulations, and those simulations are variations on simulations performed previously. And so I think that there's great capacity to learn lower cost representations of patients, and neural networks or Gaussian processes or what have you are ways of doing that with estimation of uncertainty and at very low cost. So I think these kind of machine learning emulators are going to be essential for what we're doing.
Great. And another one for Jon. Does your RBM account for heterogeneity in disease progression, e.g. pattern and rate of progression?
Yeah, so that's one of the goals of these kinds of architectures, is they can account for patient-to-patient variability in the expected progression from the data you measured at baseline. So a lot of the models that we use are Markov chain Monte Carlo models. They take the patient's data at baseline, they predict the next time point, they take that information, predict the next time point, and kind of go on through the course of the trial. And that allows you to develop really targeted disease progression predictions for each participant in a trial.
Great, yeah. That sounds good. We've got a lot of questions too. I'll ask a couple of more, and then we'll probably put the rest in the Slack if that's okay, just to keep us to time. So maybe just a question for both of you. How do you compare the methodologies for digital twin versus synthetic data generation?
Jon?
Sorry, you said versus synthetic data generation?
Yeah, synthetic data generation. Is there an overlap in methodologies? Maybe no.
I think, yeah. So certainly, yes. There are some papers where people are trying to generate recently synthetic, like MRI and CT scan data, for use in developing machine learning algorithms. I think one difference in synthetic data generation is that you're not trying to couple it to individual people, individual participants. So instead, you're trying to describe a whole population and sample from that population. I think that's probably one of the bedrock things about a twin is it is a twin of someone in the real world.
Steve, is there anything you wanted to add on that?
Yeah, I guess that there are, I think that the similarity, I guess, is also though that you can make a twin of an individual person, but then you can do what if testing. So if you needed more synthetic data about people that had a particular device or particular drug, and you can simulate that, then that does provide a way of creating an augmented synthetic data set from an existing data set, which might have a richer range of variables or a richer range of physiological conditions. So I think it could be complementary.
Perfect. And again, I think maybe a question for both of you about how do you factor in the kind of medical history of patients in your model for generating the digital twins? I guess in this sense, you've both kind of answered this in terms of you're working on the data that you have, but is there anything you'd kind of add around that point?
I think if we move to making larger numbers of twins and to more diverse patient groups, then you can start to use patient demographics to learn how to initialize priors and have more constrained priors for the parameters for your twin. And so that then means that information which isn't necessarily directly represented explicitly within your twin. So for example, we have hearts, but we have no way of explicitly representing any mental health issues. If we found that people who had potential mental health, as an arbitrary example, had particular cardiovascular phenotypes, then we start to learn the mapping between those and use those as predicting. And so that would be a way that you could start to take either comorbidities or lifestyle choices and use those to make priors for your twin.
Yeah, I think that's a really useful example. Jon, I guess also a bit more challenging with clinical trials is it's a bit more of a snapshot.
That's right. So they linking to medical records is extremely rare in trials. You can get historical information from information that's collected in a trial about comorbidities, past disease history, things like that. But it's often a snapshot of baseline. There are sometimes pre-randomization visits, like screening visits, that you can leverage for a better kind of picture of the initial trajectory of disease. But that is a challenge in trials.
Definitely. Okay, I think I will draw us to a close there. Jon, there's a question about is there kind of a source paper that can be shared for the application of the digital twins in the Abbey study? I don't know if there's anything you can share.
There is a talk we gave at AAIC in 2022, just last year. If you look, it's published in a journal, the full abstract. But that is to the extent currently. We would love to publish the results.
Perfect. Abstract sounds good for now. I know we've got a couple more questions left in the chat, so I will make sure I put those in our Turing-Roche Slack space for our speakers to answer, and you can pose any additional questions to them there as well. So with that, I just want to say thank you so much to both our speakers. I feel like that was a really great 2 examples of how digital twins can be used quite differently within the health space.